Welcome everyone. This is our NERC session on microbial investigations of microbial fitness, virulence, and resistance. We have a great lineup of speakers today across the Northeast INBRE and COBRE programs. We just wanted to give you a brief outline of how we're planning on running the program, but first I'll introduce myself. I'm Sujin No. I'm an assistant professor at Colby College in Maine. My co-chair is Sally. Do you want to go ahead?
Sure. My name's Sally Molloy. I'm an associate professor in microbiology at the University of Maine. And Dr. No and I would like to welcome you to this NERC session on microbial fitness, virulence, and drug resistance. And a big theme in this session is how various microbes adapt to their host environment or exposure to various environmental stresses like antibiotics, but also the host's ability to recognize and respond to microbes. And our first speaker today is Dr. Daniel Shultz from the Geisel School of Medicine at Dartmouth. And he's going to be talking to you today about the evolution of antibiotic responses in complex dynamic settings.
So, sorry, Daniel, but essentially, if you have a question during the talk, please type them into the chat box. When Dr. Shultz is done with his talk, he will go ahead and open up the chat as well as answer verbal questions from the audience. So please go ahead. Okay, we're good. Can everybody see it? We're good. So first, thanks Sally and Sujin for inviting me. This is great. I was very excited to when I got the invitation myself. So I'm Daniel. I'm from Geisel Medical School at Dartmouth. In my lab, we're broadly interested in the dynamics of cell processes. So we want to know how these processes, how they operate, how they're regulated, and how they evolve. And we have a heavy focus on antibiotic resistance mechanisms, which are highly dynamic. And we want to know how these mechanisms that typically originate in the soil, how they evolve to adapt to more complex environments, so to other complex environments, such as the clinic. We're a part dry, part wet lab. So we combine bench experiments with computational approaches. So just so you know what we're in for, we use a lot of microfluidics, both to study single cells and to study microcolonies. We use experimental evolution, both for single strains and for communities of microbes. And we also use computational approaches like mathematical modeling or bioinformatics. So when people tend to think of antibiotic resistance, usually people think of pretty black and white terms like, can these cells grow in the presence of the drug? But in reality, the picture is a little more complex than that. And very often, the resistance that we see in the clinic, it doesn't really match what you test in the lab. So here I'm showing you this video, this experiment to start imaging these resistant E. coli cells right when they're exposed to tetracycline. And you can see that it actually takes you a little while to notice anything. And that's because most of the cells are arrested right at the moment of when they're exposed to the drug. And it takes a little while to see that actually a few cells end up surviving and they repopulate the colony. And I actually wanted to hold on to this mental picture of you having to wait a little bit to see anything happening here, because that's going to come back at the end of the talk. So the point that I want to make here is that what happens in the beginning, right as you expose these cells to the drug, is very important, and is actually literally a matter of life and death to the cell, and also that it generates quite a bit of heterogeneity. So this is what we're going to talk about today. So first we're going to use tetracycline resistance in E. coli, which is basically the most studied antibiotic resistance mechanism in the most studied organism there is. So we'll try to see how antibiotic resistance, how they proceed under the three main sources of heterogeneity that we know in microbial cultures, which is stochastic variation, environmental variation, and genetic variation. And then at the end, we're going to look at like a real clinical example of a microbe adapting to a human host, which is Pseudomonas infections in cystic fibrosis patients. Okay, so first, stochastic variation. So if you look at a population of bacterial cells, and they're all genetically identical, and they're under the same environment, and still you see a lot of variation. If you look at the expression of a certain gene, you're going to see a lot of variation in that population. And that's because the intracellular components, especially in bacteria, they're present in small numbers. So they're subject to a lot of noise. So that makes bacterial physiology pretty noisy, and this noise can kick cellular responses in different directions. So to look at this, we have developed a microfluidic device to image single cells as they are exposed to antibiotics. So this is the design here. We have these narrow chambers where they're the width of a single cell, and we can place these cells at the top of this chamber, so they're kind of like locked in there. And then the daughter, as they grow and divide, the daughter cells get washed away in this medium channel, this feeding channel here, where we flow medium. And the cells at the top, they just stay there forever, right? So not forever, but for a very long time, and we can follow them as they grow and divide. So we have a valving layer here where we can switch between different media. So the idea here is that we're going to grow these cells on fresh medium for a while until they stabilize, and then we're going to hit them with drug and see what happens. And we can measure gene expression and cell growth as we do that. So here is resistant E. coli. We're going to expose them to tetracycline at time 0 here. And we're measuring expression of these two genes here. So the red is Tat A, is the efflux pump that kicks tetracycline out of the cell. And green is the repressor of Tat A. So I'm going to start the video here. See that the cells are growing. They're pretty happy. And then, okay, like now we expose the cells to the drug. And the cells are all genetically identical. And still you see a lot of variation here on the outcomes, right? Mostly you see that the cells that by now are still growing are these yellow ones, right? And then you see these very dim cells that were arrested pretty early on before they could express resistance. And then we also see these very red cells that they actually grow slowly for a while. But then they end up overexpressing resistance, but then they are arrested anyways. And that's because like making too much of this efflux pump is actually toxic for the cell. So that's not a good thing that they make too much of it. So how does the cell regulate these responses when they're in an environment where this drug concentration can change very quickly? So we developed a mathematical model to model this response, and I'm not gonna go into equations right now, but we use differential equations to basically include all the main biochemical interactions of this process, which in the case of tetracycline here, so tetracycline diffuses into the cell, it binds to the repressor TETR, and then TETR loses its affinity to the DNA, end up releasing expression both of itself and of Tat A, and then Tat A pumps drug out of the cell. Yeah, so then if we write this equation, then we can simulate this system, and what you see is that initially when there's not a lot of efflux pump, you have this sharp increase of drug inside the cell, intracellular drug, and they have this first peak of drug concentration, and then this drug concentration inside the cell starts going down as the cell makes the efflux pump. But then at concentration efflux pump keeps going up until the cell can shut off production. So there's a peak of efflux pump concentration, and then it stabilizes to they stabilize to some intermediate values at the end. So actually these first two peaks and the end here basically correspond to the three phenotypes that we just saw in the microfluidics, right? So the cell can die here with too much drug, the cell can die here with too much efflux pump, or it can like keep growing, right? And then another point is that the cell has some feedback mechanisms that can basically amplify the small variations, the stochastic variation in expression that we see, and take this cell responses to different outcomes. So if you consider this interplay between the influx of drug inside the cell and expression of resistance. So if too much drug comes in, then the cell cannot express resistance properly, and then that leads to even more drug coming in, right? Or on the other hand, if the cell can express enough resistance, that leads to even less drug coming inside the cell and more expression of resistance, right? So there's this positive feedback mechanism, and this is known to generate bistability, right? So you can take the small variations and actually have like divergent paths on this of these responses. So this is just a comparison here of these trajectories, comparing here. This is those fates that we see in the microfluidic experiment. That is what we get from the model. So we can actually have a stochastic model of this, that we can recover this diversity that you see at the single cell level. Another thing that I want to talk about is this environmental variation. So this is another source of variability in microbial communities, and this is because of differences in the immediate surrounding of the cells in a colony, and this can also lead to different phenotypes. And this is a general feature of structured communities where the cells exchange chemical compounds with the environment, and they can create gradients across the colony. So we have another microfluidic device to study these structured colonies. It's very similar to the one that we just looked, but the channels are larger, so we can fit the entire colony over there. Well, not entire, but like a microcolony. So nutrient only comes from the top. So then consumption of nutrient by the cells at the top here create a gradient of nutrient concentration towards the bottom. And at some point, the cells run out of food, right? So we have this somewhat sharp boundary here between fast-growing cells and at the top, dormant cells at the bottom, and like a somewhat narrow region of slow-growing cells. So this is what happens when you expose this colony to drug at the top. So so what happens here is that as you expose these cells to tetracycline, they reduce the growth of the cells at the top, and then more food becomes available to the cells at the bottom. So basically reactivate these cells further down the trap. And then during the response, like this slow-growing cells around the boundary here, they end up overexpressing resistance, just like we saw in the previous experiments. And as the cells at the top also end up expressing resistance and become resistant, this boundary between the growing and dormant cells shift back up. And what you see is that this strong expression of resistance with the cells around the boundary here. So this is a time lapse of how the response proceeds here. So at time 0 is when you add the drug, and you see that actually these cells at the boundary, they have initially slightly higher expression of resistance. They are the ones that actually expand. There's the one that grow and take over the top of the colony, right? And as the response proceeds, you see that the cells, the slow growing cells at the boundary here, they end up overexpressing resistance. And at the end, they're not growing anymore because the boundary shifts back up as the cells consume more resources. And then you create this layer of very resistant dormant cells that stays even after you take the drug out. So then this basically creates this layer of dormant cells with high expression of resistance that they stay, that basically prime the colony to be more resistant to subsequent drug exposures. So here's a chymograph. So the x-axis here we have time, and this is depth of the cell, of the trap. In blue, it's growth. We have growth, and in red, expression of resistance. And you can see here both this activation of dormant cells at the bottom in the beginning of exposure. So this is drug here. This is the period of drug exposure, and it's creation of this layer of dormant resistant cells. So this is a pretty clever mechanism of collective resistance that we see from this environmental variation. Okay, so the last mechanism of variability that we're going to look at is genetic variation. So in an isogenic cell population, this can happen because of mutations, recombination, etc. So here we're going to look at this the evolution of tetracycline resistance E. coli in different dynamic environments to see how these bacterial populations can learn how to regulate their responses according to the environment that they're in. So the tetraoperon that contains the tetracycline resistance mechanism, it's present in a transposon, and it's present in a bunch of different species. So it's mobile, and in this intergenic region here, it has a lot of overlapping promoters and repressor binding sites that implement some pretty sophisticated regulation. And if we look at gene databases for all the homologs of this operon, and look at the variation along the mechanism, we see that this region containing the regulation here has a lot of diversity. So this suggests a strong role in adaptation of this mechanism to different hosts or environments. So we have this. Other device here is a fluidic device where we can propagate continuous cultures. And so we have a growing culture here, and we can drip fresh medium and drug automatically and measure optical density. And we can control the environment that these cultures grow very precisely. And we can impose different environments on them and propagate them long term, so they evolve. And here we're going to look at what happens when we evolve them in a slow changing environment where the drug concentration kind of ramps up slowly, or another environment where once a day we just hit the cells with a high concentration of drug. So this is what the data looks like for a week of evolution. So this is drug concentration. Here this increases slowly, and here we have these peaks of drug concentration once a day. So what we were trying to see is that in this slow changing environment, basically the cell doesn't need to switch resistance on and off very quickly. So they can just adapt their expression of resistance to the condition. Well, in this case here, but sometimes we have a lot of drug, sometimes not, then the cells need to learn how to switch resistance on and off effectively. And here is what we see in the evolved populations, and that's basically what we find. At the end, both of these populations, all of these populations actually get better than the wild type, both at growing and at resistant drug. But you see that the cells that evolved in the slow changing conditions, they become more resistant at the expense of not growing so well when there's no drug, because they presumably because they keep the resistance always on. While the cells that evolve in this fast-changing environment, they don't get as resistant as the slow-changing population, but they grow very well at when there's no drug. So they learn how to switch this resistance on and off. We're currently sequencing these populations to see how this plays out. Okay. Okay, and then the last thing that I want to talk about is Pseudomonas. Oh, sorry, I'm late. Pseudomonas adaptation to the human lung. The Pseudomonas infections, they usually come from environmental strains, and half of the strains that evolve in the human lung, they develop mutations in this MACZ repressor of resistance. So here's a video similar to the first one we saw, where these strains, where you see a lot of variation in both in the expression of resistance and in their ability to survive exposure, right? And so the cells that initially have more expression of resistance, they're better at surviving. So we developed this assay where we expose growth curves. We make, oh, am I late? So okay, where we have growth curves, and we expose these cultures to a certain drug concentration in the middle of exponential phase, and then what you see is this delay in their growth until growth is resumed, right? And this delay is what we saw in that first experiment. So it's the time that it takes for the cells that survive exposure, to grow past this background of arrested cells. And from this, we can not only see how the cells grow in the presence of the drug in the steady state, but we can also estimate the fraction of the population that can survive this initial exposure. So when we look at these mutants that develop in the lung, we see that they actually do not have more resistance than the wild type in MIC, right, in the traditional measures, but they do develop a faster response that supposedly makes them more adapted to changing environments, to antibiotic treatments. And we're currently looking at a collection of Pseudomonas strains for all kinds of different environments. And as you can see, they are all over the place in terms of their resistance profile. What we learned is that there's some resistance mechanisms like the one that tetracycline resistant in E. coli. They're very adapted. They're very good at these dynamical situations. But then there's some antibiotic resistance, like the ones that you see in Pseudomonas, that they're very slow to respond. But when they evolve in a human host, they actually acquire mutations that make this response much faster. And we're trying to understand how this different how the cells can adapt their regulatory pathways to basically become better at responding to this more changing environment. So I thank my lab, Jack, Hao, David, and Mina, our funding sources. And I'm sorry if I went a little over.
We went a little bit over.
Okay.
Thank you, Daniel. And I actually had a question, and there's questions in the chat, but I think we'll come back to those questions at the end of the session.
Okay. I'll be here.
Awesome.
Okay, so in the interest of time, we will move on to the next speaker, who is Sally Molloy at the University of Maine. And Sally, you can take it away. I think you can probably just share your screen.
Daniel, can you help me transition to my?
Yes.
Great.
There you go.
Oops. All right. So my name's Sally Molloy. I'm at the University of Maine. And I actually have a background in fish health, but I work with undergraduate students. And in giving them various projects, we discovered that cohabitating prophage alter bacterial gene expression and drug resistance in Mycobacteria. Okay. So prophage are known drivers of antibiotic resistance in bacteria. And Wang showed in E. coli that removal of 9 coexisting prophage impacted the bacterium's ability to form biofilms, but also respond to various environmental stressors, including antibiotics. In fact, each of the prophage contributed in a different way to antibiotic resistance. And so these prophage collectively enhance the ability of the bacterium to adapt to environmental stressors, including antibiotics. Mycobacterium abscessus is one of the most antibiotic resistant pathogens, but we really know little about the prophage of these bacteria and how they contribute to drug resistance. It's an opportunistic pathogen that causes pulmonary and disseminating infections, particularly in people that have underlying pulmonary disease like cystic fibrosis. This is Isabel, and she had a double lung transplant and in the process acquired an M. abscessus infection that was totally resistant to drugs. And she was on palliative care before Dr. Hatfill here identified 3 different mycobacteriophage that could effectively infect and kill the strain that was causing her infection. And he's since then been working with a lot of clinical M. abscessus isolates. And these are not only highly drug resistant, but he discovered that they're also fairly resistant to lytic phage infection. And why these strains are so drug resistant and resistant to phage infection, well, there's probably multiple factors that contribute to that. But one thing that might be important is that most of these strains are lysogens. So what's a lysogen? Most of you recognize a phage as a virus that infects bacteria. It replicates its genome, makes new particles, and these are released through cell lysis. But many phage can carry out the lysogenic cycle, in which the phage genome integrates into the bacterial host genome, and that's called a prophage. And then the cell harboring that prophage is called a lysogen. And the prophage now can replicate every single time the cell divides, and it maintains the state of lysogeny by expressing at least one gene called the immunity repressor, which essentially represses all the genes needed for the lytic cycle. And so the fate of this prophage is now tied to the success of the bacterium. So it's not surprising that they've evolved to encode additional genes that contribute to the fitness of the host. And some of these genes, for example, protect the cell against superinfection, which is a major threat to the bacterium and the prophage. But some of these genes also contribute to virulence and potentially drug resistance. Now, the Hatfill lab continues to develop phage therapies for drug-resistant mycobacterial infections. And in the process, they looked at about 82 clinical isolates of M. abscessus and examined the prophages in those genomes and discovered the majority carry prophage, and in some cases up to 6 prophage. And they sorted these prophage genomes into what are called MAB clusters, A through Q, based on gene content. And along the way, they discovered many of these prophage genomes contain toxin antitoxin systems, which are known to contribute to drug tolerance, but also these really cool polymorphic toxin systems. And these are typically secreted and sometimes delivered to other cells, eukaryotic or bacterial. They have 3 genes, at least one WXG100 protein, which is like the M. tuberculosis ESAT6 protein, a large polymorphic toxin, a cognate immunity protein that prevents self-intoxication. And we don't really know yet how these types of genes impact the fitness of the host, but I'm going to come back and talk about polymorphic toxins a little bit more at the end of my talk. So Most of the time, extensively drug-resistant and totally drug-resistant isolates can be explained by acquired resistance, and that's where there's a mutation, for example, in the ribosomal RNA that prevents drugs like macrolides and aminoglycosides from binding the target and being effective. But not all extensively resistant isolates have these mutations. What all of the drug-resistant isolates share is high expression of this conserved mycobacterial transcriptional regulator called YB7. And YB7 positively regulates a large regulon of genes in M. abscessus, many of which contribute to intrinsic resistance. And that includes things like the TET efflux pump that Daniel was just talking about, but also enzymes that modify drugs and even contribute to macrophage survival in the macrophage. So the higher the YB7 expression, the higher the expression of all these target genes, and then the more resistant the cell is, right? So what turns on YB7? Low levels of drugs, intracellular environment of a macrophage. And today I'm going to be talking about how prophage can also contribute to high YB7 expression. And our story begins with an undergrad named Erica Sewell looking for an honors thesis project. A colleague and I had her look for prophages in this pathogen Mycobacterium cheloni, the Berge strain. And this is very closely related to M. abscessus. It only grows at 30 degrees, so it doesn't cause a severe disease. But it is fairly drug resistant. And Erica found a single prophage that we call McProf. And we didn't think much of McProf at the time, but over the years, we started to realize that McProf does some interesting things. And the following annotation has evolved. This is the McProf genome as it is in the bacterial genome. So it's oriented at L to at R. This ruler represents the coordinates of the genome, and the boxes above and below represent genes that are forward or reverse transcribed. And it carries integration and immunity genes, lytic genes for replication, late lytic genes for the capsid structure, lysis genes. And on the very right arm is a group of genes that we know are expressed during lysogeny. We don't know the function of a lot of these genes, but they could potentially contribute to the fitness of the host. And there's one set of genes that we think is pretty interesting, and it happens to be one of these polymorphic toxin systems. Fast forward to COVID-19, when we're looking for projects that undergrad students could carry out remotely. We, at this point, are understanding that McProf does pretty cool things. So we asked these students to look in public databases in mycobacterial genome sequences for prophages that look like McProf, and they found 25 strains of clinical M. abscessus isolates that carried McProf-like prophages. Only 4 of them are unique, and we have those aligned to McProf here up on top. This purple shading indicates high nucleotide identity between the genomes. So you can see this is a pretty conserved group of prophages. And with the help of the Hatfill lab, we sorted this group of prophages into a novel cluster, MAB-R, which is distinct from the other prophages described by the Hatfill group. And so although this is an M. cheloni prophage, it's found in quite a few M. abscessus isolates. About the time Erica was annotating the McProf genome, I had another student, Emily Illingworth, that was interested in how prophages change gene expression and phenotype of the bacterium. We didn't have a strain that was a non-lysogen, that lacked a prophage. So we decided to add a second prophage to our system. And Emily looked through a large library of phages and found 3 that could infect M. cheloni, and only one of those was capable of making a lysogen. So we made what we're calling a double lysogen that carries both the prophage BPS and McProf. And then we looked at how the presence of this phage changed drug resistance. And so we did grueling MIC assays. And what I'm showing you is viability of these cells after exposure to amikacin. We tested a number of different drugs, but I'm just showing you the aminoglycoside assays. And our strain that just carried McProf would grow at 32 micrograms per mil. As a positive control, we treated this strain with acivicin, which we knew would turn on YB7 and increase resistance, and that's what we saw. So an increase of an MIC to 64. But our double lysogen also grew at 64 micrograms per mil. And so the presence of the second prophage increased the drug resistance. We saw the same thing with tobramycin. So what are the genes that could be involved in this change in resistance? We performed RNA-seq on these two strains. And of the 4,867 genes in the M. cheloni genome, 8.5% were differentially regulated in this double lysogen. There were a lot of downregulated genes. I'm just talking about the ones that were upregulated. The majority of the upregulated genes belong to the YB7 regulon, and that includes the YB7 transcriptional regulator itself, which was upregulated 26-fold, and many of its target genes were upregulated 10-fold. And again, these genes contribute to drug resistance and survival in macrophage. I'm just going to sneak in this most regulated gene in the set was a flotilin protein that was upregulated 100-fold. I don't have time to talk about it, but I'm super interested in this gene. And if anyone knows a lot about flotilin proteins, I'd love to chat with you more. My graduate student, J.C. Cushman, wanted to understand how each of these prophages were contributing to the drug resistance and changes in gene expression. So she cured our McProf strain from that prophage so that we could have a non-lysogen strain. And from that, she created a BPS single lysogen. And we went back to doing our MICs. And here's our single McProf lysogen at 32 and our double lysogen. What was interesting were strains that lacked McProf had the lowest MIC. And so that told us that BPS isn't contributing to the drug resistance. But McProf clearly is playing an important role. The other thing we learned was that the McProf strain treated with acivicin had a higher MIC than the non-lysogen treated with acivicin. So McProf alone increases amikacin resistance. McProf enhances the response to acivicin. And it also looks like McProf enhances the response, the amikacin resistance response to phage superinfection. So again, the acivicin was a positive control, but we went back and treated all 4 strains with and without acivicin. We didn't see a big change in MIC for our double lysogen here in fluorescent green, treated with acivicin compared to the single McProf lysogen treated with acivicin, but we did consistently see an increase in cell viability. So JC then looked at YB7 expression across our 4 strains. And of course, we saw the big jump in YB7 and our double lysogen. But what surprised us was the single McProf lysogen had pretty low YB7 expression. It really wasn't very different than our non-McProf strains. And so that was our first inkling that, well, YB7 might be important for the observed drug resistance. There's probably some YB7 independent mechanisms present as well. So we know from the literature that sublethal concentrations of drugs turn on YB7 and increase resistance. We certainly saw that with our non-lysogen, but when McProf is present, that further increases amikacin resistance, as does the presence of a second prophage, so superinfection. And although not an increase in MIC, there's an increase in viability for cells that have 2 prophage and are treated with sublethal concentrations of drugs. So McProf seems to be playing an important role amplifying amikacin resistance in response to different stresses. We also wondered whether we could see a similar pattern in YB7 expression if we treated them with acivicin. And the answer to that question is not so much. So each of our strains treated with acivicin, shown in black here, had an increase in YB7 expression, with the largest response in YB7 in our single McProf lysogen. But overall, the YB7 expression wasn't very high in the strain, even though it had the second highest amikacin resistance. Again, indicating that there's other factors at play besides YB7 here. In a nutshell, sub-inhibitory concentrations impact YB7 and drug resistance, and if McProf is present, that's amplified. We now know that phage infection in the absence of things that we know turn on YB7, like the macrophage or sublethal concentrations of drugs, also turns on YB7. We're interested in whether McProf, might enhance YB7 expression or even survival in macrophage. And what we're working on right now is identifying which McProf proteins are contributing to these interactions. And we know the expression profile of McProf. We're cloning all these genes and looking for their impact on drug resistance and gene expression. But I'll draw your eye to those 3 genes that are part of the polymorphic toxin system. This is just preliminary data, and I won't talk too much about it, but I just wanted to give you a little taste. Here are these 3 genes, the ESAT6-like protein, a polymorphic toxin that's already been studied in Agrobacterium, and it has DNase activity, and it's cognate immunity protein. We cloned this into an integrative plasmid with the native promoter, and then looked at YB7 expression in strains that either had the BPS prophage or didn't. And we saw a significant increase in YB7 in our recombinant strains, only if BPS was present, but it wasn't to the levels of strains that carried the entire McProf prophage. So there's more to learn here, but we think this could be important in YB7 regulation. We have some additional data that helps us understand this a little bit better, but I don't have time to show it to you. So maybe my next question might be, do prophage encoded polymorphic toxin systems allow mycobacteria to adapt to environmental stresses? I'm going to thank this large crowd of undergraduates that have helped done this work. My graduate student, JC, and a second new graduate student, Sarah McAllister, who did a lot of this work. My mentors, Dr. Graham Hatfull and Dr. Rob Wheeler. My collaborators, Dr. Keith Hutchison and Dr. Josh Kelly, and funding from Maine INBRE.
We have time for a question, if anyone from the audience. Well, I'll ask a quick one. So when your student was doing the database mining, how often were the 2 prophages that you were looking at found together?
So BPS is actually not, I don't think we've found any prophages that look like BPS in any of the pathogenic strains. That's an organism, not an organism, that's a virus that was isolated on M. smegmatis. It was actually one of the viruses used to treat Isabel in the beginning, engineered, of course. And it infects a lot of the pathogenic strains, but we haven't seen prophages like that in any of the pathogenic strains, just in M. smegmatis. But of the strains that we looked in, we also looked at cohabitating prophages with the McProf-like prophages, and most of them carried a second prophage, but those prophages looked a lot more like the prophages that the Hatfull group described. So additional polymorphic toxins, additional toxin antitoxins in those genomes as well.
And do you want to take Catherine's question from the chat?
Okay, Catherine's question is, have you tested any of the transcription factors on McProf to see if they influence YB7 expression? See if I can digest that, any of the transcription factors on McProf. So you're asking, are there transcription factors encoded by McProf that possibly contribute to YB7 expression? I haven't identified any necessarily there certainly are transcription factors, probably, at least there's lots of DNA binding proteins in the McProf genome. I do know which ones are expressed, and I think we almost have all of those cloned, but we haven't done the gene expression assays on those yet. And as far as I know, only a couple of them are, maybe one of them is a DNA binding protein, but I don't know of any specific transcription factors yet. So I'm working on it. Great question, Catherine. So I think that, I hope we're kind of back on time. And I think what I'll do is introduce our next speaker, which is Dr. Sujin No.
Okay, so hopefully we can see this all right. I'm Sujin No. I'm an assistant professor at Colby College. I also mostly do research exclusively with undergrad students. So as we all know, microbes are everywhere, and there are many microbes that form long-term associations with eukaryotic hosts. The system that I work in, I work with both hosts and symbionts that are microbial. Anyway, so we'll call these relationships symbiotic when you have a host and a microbe that are associating with each other in some sort of concrete way, so they're not just found next to each other in some sort of lab or environmental condition. So one of the things that I'm particularly interested in is trying to understand the genomics of symbiosis when you have a host and a microbe cohabiting and they are exposed to both lots of different social conditions or environmental conditions. As with infectious diseases causing viruses such as SARS-CoV-2, the genomes of both the host and the symbiotic microbe will, of course, influence the outcome of such long-term associations. So this is a particularly cool study that came out recently where we can see in the human genome genetic loci that are responsible both for susceptibility on the bottom and then severity of COVID-19 infections. And so not only are our genomes going to influence what kind of association we'll have with symbiotic microbes, but also the relationship itself will also shape the ways that our genomes evolve. And so a lot of the research in my lab is trying to uncover these types of patterns by using our eukaryotic host, Dictyostelium discoideum. So the amoeba Dictyostelium discoideum, that we'll call Dicty for short, is a macrophage-like model. It's been used pretty successfully to study lots of different kinds of intracellular bacterial infections, such as the ones that you see highlighted in this particular figure. So amoebas have a important place in disease ecology, not only because of their occasional role as human parasites, such as the so-called brain-eating amoeba, Naegleria fowleri, but we have increasing evidence that when we have grazing amoebas or protozoa, what happens in the wild is that they're feeding on bacteria. When those bacteria can successfully invade these protist hosts, they can act then as reservoirs in which the bacteria can adapt to a eukaryotic host environment and then ultimately become more virulent pathogens to humans when they encounter us. The symbiotic microbes that we work with are 3 newly described species of Dicty symbionts. They are highlighted here: Agricolaris, Haliella, and Bonia. These guys are found in the genus Paraburkholderia that is shown here in this clade. So we described these species new to science recently. This Paraburkholderia clade that you see shown here mostly comprises plant symbionts or environmental Burkholderia sensu-lato. They are actually in a separate clade compared to our more familiar pathogenic Burkholderia that you see here used as an outgroup in this particular phylogeny. The Burk that are found in Dicty are actually only found in roughly about 25 to 30% of all wild Dicty. And so this figure here is showing you across a bunch of different wild Dicty isolate populations, the proportions of the 3 different symbionts that are found in lots of different host strains. And so these color-coded boxes indicate how often you see these different types of symbionts. So across all of these, you can see that there's quite a bit of variation in how often these symbionts co-occur with their hosts. Like the other ones that I showed in the figure previously, these are intracellular symbionts. And these Burk are also the only ones in that phylogeny that I just showed you that can persistently infect hosts. So they can form a facultative intracellular symbiosis. This means that we can actually isolate and culture these Burk outside of their host. We can then infect Dicty that are naive or originally found without any symbionts themselves by placing them in the same petri dish, essentially. And so this makes it really convenient to do experiments that, like the ones that I'll talk to you today, where essentially mixing and matching hosts and symbionts to try to better understand the circumstances that led to the co-occurrence patterns, for example, and any variation in genomes that we've been seeing. So experiments done in the system so far indicate that native hosts that come isolated with their own symbionts versus naive hosts that don't actually respond differently to infections that we can create in the lab. So for example, in comparison to the native hosts shown here and the naive hosts shown in the gray bars, when you have control conditions without any infections, there is no difference in spore productivity or how many spores these amoebas make, versus when you create infections in the lab, naive hosts suffer a bit when they are infected with these symbionts. Burke itself also has varying fitness outcomes when it is inside versus outside of a host. So in this particular study, you can see both the orange points and lines and the blue points and lines. So the orange points are when Burke is inside of a Dicty host. And in the Agricolaris species versus in the Haliella species of Burke, you can see that there's a difference in terms of whether Burke being inside of a host is actually experiencing higher fitness compared to when it's on its own. So we see this variation in host fitness and symbiont fitness when you see either the symbiosis for when each of these hosts and symbionts are on their own. Ultimately, my goal with this particular INBRE project is to be able to identify genomic factors in both the host and the symbiont that may determine these variable fitness outcomes. In addition, I'm very much interested in trying to figure out an explanation behind why not all Dicty in the wild are infected with Burke, because in the lab, we can easily induce symbiont infections. Like I was saying, you can essentially put a Burke on the same petri dish as amoebas and they will immediately become infected. And so, for example, is this because the native Dicty hosts have a higher specific tolerance to the Burke that they are found hosting, right? This is my hypothesis as to what might be going on in the natural system. As a lab, we are also interested in understanding the Dicty-Burke system as a way of looking at social infections. So close social interactions have been shown to predict more similar microbiomes and vice versa. There have been several model systems that include humans, baboons, and also more recently honeybees that show a lot of this work. The social cycle of Dicty makes this a really great system to think about social symbiont transmission. So when you have amoebas in the wild, they are in usually forest soils and feeding on bacteria. When these bacteria become scarce, usually we think it's because of seasonal fluctuations in temperature that make it so that the bacteria aren't growing quite so rapidly. The amoebas begin to starve. They secrete a cyclic AMP signal that causes them to aggregate and form these multicellular mounds. They then develop into motile slugs that then ultimately become these tiny fruiting bodies. And these fruiting bodies are visible, so they are somewhere not quite at the 2 millimeter height range. When you have these fruiting bodies, we can then collect the sorus or the top of the fruiting body and then see what spores are present and whether there are any symbiont infections in here too. And so these social cycles are really interesting because they ultimately force hosts to be in close proximity to each other and then can present an opportunity for potential hosts to be exposed to Burke infections if there are any native hosts that are in this mix. So these are all unpublished data that we just collected over the past year and into this summer. A lot of these experiments, so far we have only been doing it with one Burke species, Paraburkholderia bania. So we designed a two-part experiment to look at both host fitness and symbiont fitness, and we're going to look at the symbiont part first. So to try to address the genomic predictors of host-symbiont interactions in the system, in this first part, we're essentially asking the questions, does symbiont horizontal transmission actually occur during social cycles? And then what affects any variation in transmission if we are able to see this? So we used 3 strains, each of Dicty as well as of Bonnia, our Burke, and a full factorial design. So unfortunately, this took quite a while to be able to collect all these data. And to start this experiment, what we did is we initially infected Dicty hosts at different multiplicities of infection with RFP labeled Burke so that we could visualize these later. Then in the experiment, we labeled a population of uninfected Dicty of the same strain with a membrane dye so that we could also see these. They would then be green. We combined these 2 populations together in a 1-to-1 ratio, and then place them on a filter so that they would begin developing and enter the social cycle. If horizontal symbiont transmission is occurring, at the end of the experiment, we would be able to detect through flow cytometry green Dicty hosts that were hosting red Burke inside of them. So to kind of give you an idea or a framework to think about the figures that I'm going to show you next, let's talk a little bit about host defense mechanisms. So largely we can classify the defense mechanisms that are occurring in a host in response to any sort of infection into 2 large classes of resistance and tolerance. So when we have resistance, this is when a host is trying to reduce or potentially completely clear the intensity of an infection. So you tend to see variation along the infection intensity axis. When you have tolerance, the host is trying to diminish the effect of having the physiological effect of having an infection. And so you see variation in slopes of infection. So you can also have both of these patterns occurring at once. So in this particular example here, we're seeing 2 different genotypes of hosts that are responding to a single genotype or a strain of pathogen or symbiont. The figures that I'm going to show you next, essentially what you might look out for are, is there any difference in infection intensity? Is there a difference in slopes? Or is there potentially both going on? And being able to classify these different mechanisms will help us better interpret any genomic differences that we detect later on as to what is going on when we have these different interactions amongst hosts and symbionts. The results I'm going to show you next are, of course, a bit more complicated because we're looking at 3 different host strains and 3 different symbionts at the same time. But I've tried to simplify it and by showing you just the ones that seem to be statistically significant differences that we can detect from the experiments. So for the symbiont transmission part, what we first discovered is that for the 3 naive hosts that we used, which are QS4, 17, and 18 here, there is always a higher intensity of BB859 infection. And so this hints at a resistance mechanism in action that may be in common across all of these naive hosts, where all of the hosts are potentially more resistant to these guys, 395 and 433 strains compared to the 859 strains. When we look at the relationship between infection intensity and then the frequency of transmission, what we see is, of course, first of all, that horizontal symbiont transmission is actually happening during the social cycle, consistent to what is expected for horizontal transmission in general. When more infected amoebas are present initially, you see more transmission occurring as well. We know that this is horizontal transmission because when amoebas enter the social cycle, essentially no one goes into cell division again. So there's very little vertical transmission that's happening, if at all. The other thing that we see here is these different slopes. So we recover the same difference in the frequency of infection that we saw in the previous slide, but we're also seeing different slopes. And so there seems to be a combination of both host resistance and tolerance that is operating here. These slopes are consistent across the different host strains. And so it seems that these effects of symbiont transmission are occurring across all of these host strains pretty equally. Okay. So next we looked at host fitness, and this will help us get a better understanding of what happens when we have newly infected hosts as a result of symbiont transmission. To do this, we did a very similar experiment, but we were essentially looking at hosts, exposing them to these different MOIs of Burke, and then checking their fruiting bodies to see what were the infection frequencies that occurred, and then how many spores are we able to count in these fruiting bodies as a measure of host fitness. So what we found is that the frequency of infection for the same MOIs was different by host. This lower box here that you see is the host strain QS17. It was always infected at a lower frequency compared to the other host strains. And then there's an interesting thing about this figure that I'll come back to, where in the previous experiment, no one was getting infected to intensities as high as the ones that we see here. So there's a difference that we see that turns out to be quite interesting, and we're trying to understand this result as well. When we looked at the relationship between infection intensity and the host fitness effects that these infections have, what we see is that the slope for BB859 is much shallower compared to the slopes for BB395 and 433. And so, as you can see, these data are quite noisy, and so we're going to redo some of these experiments to try to see if these results are consistent. But one thing that I do want to point out here, that it's exciting to see evidence of a transmission virulence trade-off, where if you think back to the first experiment where we were seeing that BB859 was infecting at higher intensities and also transmitting at higher frequencies, this shallower slope indicates that it is potentially having a less severe effect on, or a less negative effect on, the naive host that it is infecting. So the part that I said I'd come back to is this observation that we had of where the cycle ones that you see here are from the host fitness experiment, where we immediately exposed hosts to the different Burke and saw what kind of infection intensities we're getting. The cycle 2 experiments are from the symbiont transmission experiments, where we took those infected hosts and then put them together with uninfected hosts and had them go through a social cycle again. And what you can see, especially at the higher MOIs, is that when you have an infected population that goes through an additional social cycle, it is going to see a drop in infection intensity. And so this suggests that there is mortality due to infection, and also potentially some sort of incomplete vertical transmission, where these infected hosts are not always transmitting their symbionts to their daughter cells. And so we are I'm collaborating with a computer scientist, where we've been trying to model some of these factors that we can't easily measure in the lab. And so this is one of the things that we're trying to figure out, like what are the contributions of vertical transmission versus the horizontal transmission that we can measure in the lab that contribute to some of these patterns that we see when you have these dynamic social populations of amoebas in the wild. To summarize, we find evidence for a symbiont genotype that's affecting symbiont transmission, and then for host fitness, evidence for both the host and the symbiont genotype being important for the types of patterns that we see. So this is the next step of this experiment, where we are going to be adding in native hosts to the mix. So in this mock-up of what these types of patterns might look like when you have different combinations of hosts and symbionts, we have the naive hosts that we tested up top, and then the native hosts on the bottom. And so my hypothesis is that when we have native hosts to be able to compare what their fitnesses and symbiont transmission patterns are like when we have these similar types of experiments, is that if the native hosts in fact are the more tolerant hosts, for example, their fitness consequences should be much more minor compared to the naive hosts that we were looking at. And depending on what kinds of symbiont transmission patterns that we see coming out of those native hosts, we'll be able to gain some really interesting insights into what is happening with this particular system, and then hopefully extrapolate what those results might mean for commensal bacteria, etc. And so I'd like to thank my funding sources. I actually don't have pictures of all of my students that contributed to these projects, but these are the ones that I'd like to thank in particular. So yeah, that's the end of my talk.
Great, thanks, Sujin. I actually have a question, but I'm gonna save it for the end of the session, Sujin.
Okay, yeah, no problem.
And keep us on time. Let's see.
So our next speaker is Vicki Jeffers from the University of New Hampshire.
So hi there, everyone. First of all, thanks so much to Sujin and Sally for allowing me to share my work with you all today, or our work with you all today. I hope you can see my slides. They've jumped around on the screen a little bit. Let's see. There we go. Okay. So I'm an assistant professor at the University of New Hampshire, and I'm going to share with you today a story that we're just getting ready to publish, and we're just wrapping up, so any feedback would be most welcome. So the parasite that I work on is called Toxoplasma, or Toxo for short, and it's a eukaryote, so very similar to our human cells. It contains all of the eukaryotic organelles that we have in our own human cells. And as a pathogen, that presents us with quite a challenge for identifying unique targets for drug development and therapies to treat this infection. And it's an obligate intracellular parasite. It must invade and infect a host cell to survive, so it cannot replicate without its host cell. And Toxo is kind of unusual as an obligate parasite, where it's pretty clever at infecting almost any nucleated cell from a warm-blooded animal. So we see Toxo in pretty much any warm-blooded animal, livestock, birds, and it manages to infect about 1/3 of humans worldwide. So very, very prevalent infection in humans. And it has some very famous cousins. If you've never heard of Toxoplasma before, I'm sure you've probably heard of Plasmodium, which causes malaria. It's also in the same phylum as Toxo. Cryptosporidium and cyclospore diarrheal diseases that affect humans, and INBRE, and there's a number of other infections that affect farm animals and birds. So these guys are culprits for a lot of disease in both humans and agricultural animals. But the nice thing about Toxo, compared to some of its cousins, is that it's relatively easy to culture in the lab. It's not fussy, since it can invade pretty much any cell. We can grow it very easily in the lab. And thankfully, its haploid genome makes it very easy to genetically manipulate. So Toxo, for a long time, has been considered kind of the model organism for this nefarious bunch of organisms in the apicomplexan phylum. And generally, as well as a human pathogen in its own right, it's often used as this kind of model for testing different things that may also happen in its cousins in the phylum. So Toxo can be transmitted to humans in a number of different ways.
Can I interrupt you for a second?
Sure, yeah.
The slides aren't advancing.
Oh, it's advancing on my screen. Let's see.
Oh, shoot. Oh, it just did it. It did it.
Oh, it did it. Okay.
Yeah.
Let's
yeah, do
see.
you want to? Huh.
Is it full screen again now?
We're not seeing the
It's not
presentation
full
view.
screen.
Let's see. If I do this, can you see it now?
Do that same option and hit primary monitor.
There we go.
Did that do it? Did that do it?
Does that work?
No, we're still seeing on this slide, whatever this monitor is named, that's what we're still seeing.
So this was the monitor that I have. This is what I had originally. I'm looking at 3 different screens, so it's kind of silly. I've got my laptop and then 2
oh, okay.
external screens.
So does the full screen,
take this off.
Because you can change the setup that the PowerPoint screen is on, or you can share the screen that your PowerPoint went to, if your PowerPoint went to a different screen.
Oh, okay. Okay, so I'll stop share. Sorry, everyone. Okay.
And then when you click share screen again, it should come up with like different tabs. Yeah, different screens to share.
Let's see. Gotcha. Okay, let's do this then. All right. How's that now? Of course, no, wait, I know what's wrong. There you go. Perfect. Okay, thanks for that, sorry. All right, so I'm gonna have to move my computer a little bit. I might not be looking at the camera completely, but okay, where was I? Okay, so transmission of toxo can occur through a couple of different routes. So the cat is the primary host of toxoplasma. So this is where sexual reproduction takes place. And the cat then, the infected cat will release these oocysts into the environment, and humans can become infected through consumption of the oocysts through fresh vegetables that haven't been washed, contaminated water. And people can also become infected through consumption of undercooked meat from infected livestock that contain these dormant stages called bradyzoites. And so this is particularly worrying or a major concern if a woman becomes infected for the first time during her pregnancy. Those parasites are able to then cross the placental barrier and infect the developing fetus, and that can have some really serious side effects for the baby. So really, that's why you may have heard of pregnant women being told not to change cat litter. It's because of that, the chance of that infection. So when a person becomes infected, they generally experience a very mild acute infection. That's these tachyzoids are the stage responsible for this acute infection. And if they're healthy and their immune system is functional, those parasites, those tachyzoids, very quickly transition into the bradyzoid stage, which is responsible for the dormant or chronic infection. And this is the stage that's responsible for this really high human prevalence, which is about 25 to 30% here in the US. It can be up to 90% in certain regions. So really, really prevalent infection within people. And so these bradyzoites form tissue cysts. They really like to persist in the central nervous system. So you'll see a lot of cysts in the brain, and the cardiac muscle, and often the optic nerve is also a hotspot for bradyzoid cysts. And unfortunately, this stage is impervious to any drug treatment that we currently have. So this is a lifelong chronic infection. As a person's infected, that they'll carry this infection for the rest of their lives. And in general, we've considered this as a fairly benign infection. Really, we don't see a lot of disease associated with this chronic infection, but there is an emerging link now to a correlation between chronic toxoplasma infection and certain mental health disorders or behaviors in humans. So obviously this is a serious concern, and we're trying to get to the bottom of this in humans now in the last couple of years. This will stay the case if your immune system continues to keep the parasites in their dormant stage, but if the immune system stops working, if a patient becomes immunocompromised, those parasites will activate very quickly and start replicating within the infected tissues. And of course, if it's in the brain or the cardiac muscle, as you can imagine, if those parasites are proliferating and destroying that tissue in those locations, it can very quickly lead to some serious consequences. And so we have drugs to treat this stage. Those drugs are problematic. They can be toxic and have a lot of side effects or allergenic effects in some often very vulnerable patients. So of course, we're on the lookout now for some more effective and safer therapies for toxoplasma. And so this process, because we don't have any treatments for the chronic stage of infection and not great treatments for the acute stage of infection, we're really interested in studying the molecular mechanisms underlying this transition between tachyzoids and bradyzoids to try and identify therapies that we can use to treat both stages, or indeed take someone who's chronically infected, wake those parasites up and change them all back to tachyzoids, so then we can hit those tachyzoids with the drugs that we currently have. So my lab's really interested in studying how this process occurs. And one of the aspects of control that we're really interested in is regulation of gene expression. So Toxo, as a eukaryotic organism, has very similar pathway for regulation of gene expression that we have in our own cells. We certainly have this similar chromatin structure. We have RNA polymerase that's recruited to promoters due to recruitment of DNA binding proteins and modification of histones. But there are some very divergent features of this process that we think are really interesting to investigate more closely and could potentially lead to some really nice drug therapies for toxoplasma. And so one of the processes I've been studying through my postdoc and now in my lab is the process of histone acetylation. So this is the process of modification of the histone tails, where the acetyl group is added by acetyltransferases, and that opens up the chromatin, and that's always associated with activation of gene expression. So it allows the transcriptional machinery to access the DNA and transcribe the mRNA. And one of the features of this whole pathway is the role of the bromodomain. And so bromodomains, it's not an enzymatic domain, it's literally just a reader domain, where it's the recognition module or reader protein for the acetylated marks on histone tails. So this structure of a bundle of alpha helices forms a pocket that binds the acetylated lysine on the histone tail. And what's really nice about bromodomains and this recognition pocket is that it could also be potentially a really nice target for developing small molecule inhibitors to occlude the acetylated lysine and prevent that bromodomain from functioning or recognizing that acetylated histone. So this is the family of proteins that we've been really interested in studying in Toxo for the last couple of years, and with the view to hopefully developing small molecule inhibitors to treat toxoplasma. So bromodomains in Toxo, there are 12 in the genome, and we have about 6 that look like bromodomain proteins that we have in our own cells in humans that we know the function of. So in general, as a parasitologist, we're not super interested in these because they're so similar to our own cells, but we're really interested in these 6 that are kind of arbitrarily BDP1 bromodomain containing protein 1 to 6. And you can see these numbers here on the left, the relative fitness score. These are a CRISPR screen was performed to assess the essentiality of every single gene in the Toxo genome. And they were given an arbitrary score between -6 and 2. So anything closer to -6 is really essential. So you can see from these numbers that many of these bromodomain proteins are actually very close to being essential, required for parasite survival. So again, this makes them really attractive drug targets. And so one of the first experiments that I ever did when I started looking at bromodomains was really to determine if they could be targeted by a small molecule and if bromodomains are really essential when it comes down to looking at parasite replication. So IBET 151 or IBET is a small molecule. It was one of the first bromodomain inhibitors to be reported. And so I just tested the effect of IBET on parasite growth. And so here along the top, you can see the results of a plaque assay, which is how I tested parasite proliferation. So the parasites are growing in a monolayer of human fibroblasts, which you can see here. This is an uninfected monolayer stained with crystal violet. And when the parasites infect the monolayer and start replicating, they really start disrupting, they're lysing the cells and clearing the monolayer, the cells from the monolayer. So when you stain with crystal violet, you see this opening up of gaps in the monolayer that you can then use to quantify parasite proliferation. So you can see in increasing concentrations of the drug, we see a decrease in parasite replication, which was really promising to us as a potential lead compound for inhibition of toxoplasma. And so we decided to start looking for the target of IBAT in toxoplasma, and we looked at the targets in humans, and we found that IBAT pretty specifically targets this group of bromodomain proteins in humans called the BET or BET family of proteins, and there are 4, and they all share this domain topology of 2 N-terminal bromodomains, and with this NET domain or N-terminal extra-terminal domain, which is involved in protein-protein interactions. And there's a similar conserved group of proteins in Saccharomyces and yeast as well. So, and these in both humans and yeast are generally involved in recognition of the histones and initiation of gene transcription. So we took a look back at our list of bromodomains in toxo, and you'll notice here popping up right away, which we didn't detect as a human-like bromodomain protein by sequence homology analysis. We really, based on domain topology or architecture, this was the one that jumped out to us first as a potential target for IBAT. So we asked the question, does IBAT target BBP? And we had this really great reagent from GSK they shared with us where it could be linked to beads. So we have IBAT here linked to some beads, and we could test IBAT binding. And so this was our IBAT protein. It did not bind to beads alone. We saw IBAT binding really strongly to the BDP3 protein. So the BDP3 protein did not bind to the beads alone. It bound really strongly to the IBAT-coated beads, and then it was competed away with excess IBAT in the medium. So that told us really kind of conclusively that BDP3 is binding the IBAT, and we were able to show with recombinant bromodomain that the IBAT was binding BDP1. So we then tested the ability of BDP1 to bind acetylated histone, which indeed it does. We saw that the bromodomain 1 from BDP3 bound acetylated histone. And when we added some IBAT into the mix, we saw that IBAT, the drug, inhibited binding or recognition of acetylated histone, indicating that the drug is inhibiting BDP3 recognition of acetylated histones. So then we wanted to know what the biological function of this protein is in toxoplasma. So we know it's a target of this molecule, but we really don't know what it does. So our questions were, is BDP3 associated with epigenetic or transcriptional regulatory complexes? And if it really is, recruited to the chromatin at specific gene loci. And to do that, we added an endogenous tag on the protein. So we were then able to perform the subsequent experiments that we needed to do. We found that, let's see, there we go. So we found that the protein indeed localized to the parasite nucleus where we would expect. And so we wanted to perform co-immunoprecipitation to identify the complexes or proteins that BDP3 was associated with, and also do chromatin immunoprecipitation and sequencing or ChIP-seq to determine where in the genome that this BDP3 or its complex was binding to. And we're hoping that this would provide us with clues as to BDP3 function in the parasite. So our co-IP showed us that BDP3 really nicely and consistently associated with a whole bunch of these TAPs or TBP associated factors. And they all hang out together in this one complex called the TF2D complex, which is part of the complex that recognizes sequences within the promoter region. In humans, it's the TADA-box binding protein that recognizes the TADA-box. We don't have TADA-boxes in toxo promoters, so we're really not sure what sequence that this complex is recognizing. But this whole group of proteins somehow associate to the promoter region of active genes to ultimately recruit RNA polymerase to the transcriptional initiation sites. So that suggested very strongly to us that BDP3 somehow contributes to transcriptional initiation in toxoplasma. And so we would expect when we do chromatin immunoprecipitation that we would observe BDP3 binding right around here, around where transcription begins at active genes. And so when we did ChIP-seq, that is indeed what we observed. So this is just one snapshot of one of the replicates of our ChIP-seq. We found really nice BDP3 peaks right upstream of a number of active genes, but not all active genes. So this gene also, when we look at markers for transcriptional initiation and transcriptional elongation, we can see that both of these genes, this AMA1 and this hypothetical protein, are both active, and they have marks in the transcriptional initiation site for active transcription. They have marks for transcriptional elongation, but we don't see a BDP3 binding site within this gene. So we ultimately observed BDP3 binding to about 800 genes within 3 kb of the start codon of translation. So we suspect these are under BDP3 regulation, but we really don't know yet what dictates BDP3 recruitment or preference for certain genes over others. But when we did look at the BDP3 target genes, we found, really it was a mixed bag in general, but when we did go analysis of this group of genes, we found really strong enrichment of translational proteins, and in particular ribosomal proteins. So maybe this segues very nicely into the next talk, I'm not sure, but it looks like BDP3 is a regulator of ribosomal proteins amongst some other things as well. And that probably explains why this protein is so essential to the parasite. So what we know so far is BDP3 seems to be kind of a cryptic member of this BET family that we see in humans and yeast. It's targeted by IBETs. It has that similar domain architecture and is associated with a complex of transcriptional initiation. We really don't know what the second bromodomain is doing. We weren't able to observe any histone acetylation recognition function of that second bromodomain, so we're still working on figuring out what that might be. We don't know what histone mark BDP3 really prefers to bind in vivo. And we don't know if DNA binding proteins are involved in that selectivity of BDP3 binding for certain promoters. And then we're interested in its association with the transcriptional initiation complex. Of course, since BDP3 wasn't observed at all active genes, we don't think it's a core component of this particular initiation complex. So we're really curious about what else might be happening at other promoters. And so we're really interested now in this crosstalk between the epigenetic factors, which is what historically I've always studied, and now how they talk to, or this is kind of, we've shown now for the first time how those epigenetic factors interact directly with the transcriptional machinery. So we're a little more interested in this process and the conversation between the two different mechanisms. And so I'm really grateful to everyone in my lab. They're a really wonderful group of people to do science with every day. And also the Hubbard Center here at UNH, which are great. You know, we've got some really exciting things coming up with them. And this work was started in my postdoctoral lab at IU School of Medicine in Indianapolis, and Bill Sullivan was my mentor there. I'm grateful to the folks at GSK for sharing a lot of those IBET reagents originally that helped us really get to the bottom of the target of IBET. And of course, SIBR here, the COBRI supported program here at UNH for my support as a project lead. And so I'm happy to answer any questions.
I'm afraid we probably should move on to the next talk. And so as with previous speakers, we'll try to accumulate questions at the end of the session. So please stick around if you're interested in talking to them.
I had a question, but I was typing it too slow, so I'll get it into the chat box eventually. Our next speaker is Catherine Ramsey. She's an assistant professor in cell and molecular biology at the University of Rhode Island, and she's going to be talking today about ribosomal proteins and control of virulence genes in the human pathogen Francisella tularensis.
Thank you so much for that introduction. Can everyone hear me okay and see the screen all right? Excellent, excellent. So again, thank you to Sujin and Sally for organizing this. I'm really excited to be here today and to tell you about some of the work that we're doing. So I'm pleased and honored to be a recipient of an Early Career Development Award from the Rhode Island-INBRE. And again, we're at the University of Rhode Island. So our project I'm going to be describing to you today is focused on ribosomal proteins and control of virulence genes in the human pathogen Francisella tularensis. So I'm going to start off by kind of reminding you of some salient aspects of ribosome biology, introducing you to this pathogen that you may never have heard of, Francisella tularensis, and then we'll get into it. So here's my cartoon version of a ribosome. Now, ribosomes are complex, right? There are over 50 proteins, 3 ribosomal RNAs, but you're going to see ribosomes depicted in this cartoon as this 30S small subunit and 50S large subunit that come together to make the bacterial 70S ribosome. Now, the job of a ribosome, of course, is to translate a messenger mRNA into a protein. And when we think of ribosomes doing this job, we generally think of them as homogeneous, constitutively functioning machines. And what I want to point out to you today is that we've known for a long time that ribosomes are actually not homogeneous. There are a number of ways in which ribosomes can actually be quite heterogeneous. And the way in which I'm going to just talk about that today is describing alternate homologs for ribosomal proteins. So actually, most bacteria encode more than a single homolog for at least one of their ribosomal proteins. And so you can imagine that if a ribosome incorporates different homologs for a particular protein, this may change the structure of the ribosome and potentially even the function of the ribosome, right? And this is an open question in bacteria. This isn't particularly well understood, and some preliminary, some early results are indicating that in fact, changes in ribosome composition can in fact influence translation and regulate gene expression. And so I'm going to tell you a bit of a story about this today in relation to my favorite organism, Francisella tularensis. So here you can see these bacteria, which are colored in blue. Can you see my pointer, by the way? Any feedback on that? Okay, great. Thank you. So these bacteria colored in blue have replicated, obviously, at a high number within this macrophage, which is in yellow. Now, these are the gram-negative facultative intracellular pathogen, Francisella tularensis, and they cause a disease. It's not very nice. It's called tularemia. And this disease does have the potential to be fatal if left untreated. Not only that, it has fewer than 10 organisms can constitute an infectious dose. So it is, in fact, considered a potential bioweapon for these reasons. Now, we don't work with bioweapons in my laboratory, happily. Instead, we use an organism called Francisella tularensis, subspecies Halarctica LVS, or the live vaccine strain. So this is a strain that, while it's attenuated for virulence in humans, retains pathogenicity in animal models. So it's a great way to study virulence of Francisella species. Furthermore, the ability of these organisms to function as a pathogen is strictly dependent on their ability to survive within this niche, within host cells, and in particular within macrophage. And so this intracellular growth is essential to cause disease. And so we can assess virulence quite nicely in the laboratory by assessing the ability to grow within macrophage. Now, the question that's been driving much of my career is really, how are the genes that are necessary for intracellular growth regulated? And so I came to my interest in ribosomes when I came across a particularly interesting observation. And that is that Francisella tularensis only has a single homolog for most ribosomal proteins, but for a particular one, it encodes 3 distinct homologs about this protein called BS21. And I'm going to tell you why I find that so interesting. So we don't know a heck of a lot about BS21's function, but we do know that it's involved in translation initiation. And in this structure here, this is the 30S subunit stripped away of all of the proteins. So you basically have the 16S ribosomal RNA and just BS21 depicted. And you can see it's right next to this mRNA exit channel. So this is where our mRNA would exit after translation. It's also the location where you would see 5' untranslated regions interacting with the ribosome during initiation. So it's in the right spot to be controlling translation initiation, and folks have found that it is important for translation initiation. It doesn't seem to be essential. There are a number of organisms that actually don't even encode BS21 homologs, but in those organisms that do encode it, there are specific phenotypes when it's disrupted. So in Bacillus subtilis, you see changes in motility and biofilm formation. In Listeria monocytogenes, we see differences in acid stress resistance. There are differences in antibiotic resistance in Staph aureus, and in Francisella tularensis and Burkholderia pseudomallei, it's been implicated in virulence. And so this raises this question, right? Francisella tularensis has a very small genome. It's an intracellular pathogen. It's lost whole biosynthetic pathways. So why do you have multiple BS21 homologs? And what is the molecular mechanism for regulating all these phenotypes? So this raises the possibility that within a single cell, you have multiple different types of ribosomes that could contain either the first homolog, the second, the third, or maybe no VS21 at all. And so this kind of raised this hypothesis. So this is the hypothesis we've been investigating in this project, and that is that maybe incorporation of these distinct VS21 homologs can produce specialized ribosomes. That is, ribosomes that are distinct not only in structure, but also function, that may be regulating gene expression at the level of translation. So maybe VS21 is functioning almost analogously to a sigma factor for RNA polymerase. So we don't have much time. So I am going to skip over some of the early preliminary data and let you know that we were able to isolate ribosomes from wild type Francisella tularensis, and we found more than one of these VS21 proteins present in these purified ribosomes. And furthermore, we found that the second homolog was the most abundant homolog in these ribosomes. And so we then wanted to ask, so we've basically been focused on studying VS21-2 as that most abundant homolog in the wild type ribosomes to understand what its function in these cells are. So again, this hypothesis is that maybe VS21 is functioning to control gene expression at the level of translation, not transcription. And so in order to investigate this hypothesis, we wanted to do some whole genome comparisons. We took a systems level approach and compared wild type cells with those lacking VS21-2. And we performed RNA-seq to look at transcript abundance and whole cell proteomics to look at protein abundance. And so I'm plotting on this chart the log2 transformed fold change of RNA on the x-axis and the log2 fold change transformed protein abundance on the y-axis. And each dot will represent an individual gene. And these dots are colored if we detected a significant difference in one of these analyses. Now you can see that there are a number of colored dots. There are about 250 of them. And so we can detect significant changes in both transcript and protein abundance in either green or in purple, changes in only transcript abundance in blue. And you can see the majority of changes in these cells are at the level of protein abundance. So 166 genes have changes in protein abundance, that cannot be explained by changes in transcript abundance, right? And so this suggests to us, this is consistent with the idea that VS21 is controlling gene expression at the level of translation and not at the level of transcription. So this is particularly interesting to us, and we were kind of interested in understanding further which genes are being controlled by VS21-2. So if we look at these genes down in the bottom here, a number of them caught my eye. So these are the genes which would be positively regulated by VS21-2. And they caught my eye because as a Francisella researcher, I was very familiar with them. They're genes encoded on what's called the Francisella pathogenicity island, or the FPI. So this is a set of genes in Francisella species, and the live vaccine strain is 16 genes. It's absolutely critical for virulence and intracellular survival. So they encode this type 6 secretion system. And we know actually quite a bit about how this is regulated at the level of transcription. But no one had previously identified any factors that are important for regulation of translation. And so between those mass spec analyses, and then we actually have specific antibodies to a number of these proteins, we're able to validate that 12 out of these 16 genes have significant changes at the level of protein, but not transcript abundance. So again, this suggests that VS21-2 is somehow controlling expression of these virulence genes at the level of translation. So if VS21-2 is controlling virulence gene expression and we see these reductions in these proteins, we wanted to ask the question, well, does this have an effect on their ability to grow within macrophage, right? And so we can test this using our intermacrophage growth assays. So what we have here on the y-axis are the number of bacteria we recover after 24-hour intermacrophage growth assay. So about 100 bacteria enter these J774 macrophage-like cells. You can see our wild-type bacteria grow quite well inside these cells. Now, cells lacking VS21-2 have a significant decrease in their ability to survive and replicate in macrophage. So we see about a 10-fold decrease in their ability to survive, and this can be complemented by ectopic expression of VS21-2 on a plasmid. So what we've shown so far is that VS21-2 seems to be controlling gene expression at the level of translation, particularly virulence genes, and that these cells lacking VS21-2 have a defect in their ability to survive within macrophage. And so again, our hypothesis is that this is happening because of changes in the ability of the ribosome to translate particular messages, right? And so we have some preliminary data looking at kind of the molecular mechanism there. But one question about that is, if cells with different BS21 content are able to translate different mRNAs in different, kind of regulate the translation of these different mRNAs, you would imagine that you might see a structural difference among these different ribosomes. And so we want to use some chemical biology in order to probe the structure of ribosomes with different BS21 content. And so we're quite lucky because there are lots and lots of antibiotics that target the ribosome. And so we can use antibiotic sensitivity as a proxy for understanding how well or how different ribosomes between different cells might be. And so first we have to have some strains that have that are homogeneous for BS21 content. So we created these strains. So in our wild type strains, we have genes encoding all 3 homologs. And we created isogenic strains where all 3 native genes are deleted, but we have a single gene encoding 1 homolog at a neutral location. So either the first homolog, the second homolog, or the third homolog. And so then we can take these strains and ask, do we detect differences in susceptibility to antibiotics that would be indicative of changes in ribosome structure? Now, first we tested susceptibility to a DNA gyrase inhibitor, ciprofloxacin. So this was just a test to make sure that we don't have massive changes in permeability or efflux pumps. And luckily we didn't see any differences there. And then we tested 3 other 30S ribosome inhibitors, tetracycline, kanamycin, and streptomycin. And we see no difference in susceptibility to these drugs. However, when we look at a 4th drug, kasugamycin, we see a significant difference in susceptibility. So what I'm showing you here is the results from a disc diffusion assay. So on the y-axis, you have the zone of inhibition for kasugamycin, and you see that cells containing only BS21.1 are much more resistant than cells containing only BS21.3. So this suggests that actually there are some differences in structure around the location of the kasugamycin binding site. So what's really neat is kasugamycin is the only one of these drugs that specifically inhibits translation initiation. So this is somewhat consistent with our model for how this may be working. And furthermore, the kasugamycin binding site, at least on the E. coli structure of the ribosome, is fairly close to where BS21 is located. So these are all consistent with the potential for incorporation of different BS21 homologs to change or modify the conformation of the ribosome locally at that location. So this is a great way to look at ribosome structure, but it's obviously very indirect. So we wanted a more direct way to look at this. And we were able to collaborate with some wonderful folks down at Duke University, Dr. Maria Schumacher and her graduate student, Brady Travis, and they have helped us initiate structural studies of the French cell toleransis ribosome. So we purified ribosomes and sent them samples. And these are some images of some particles that they were able to generate from our samples using cryo-EM. And using cryo-EM, they were able to generate this preliminary map of the French cell toleransis ribosome, which is extremely exciting to me because this is the first map of the French cell toleransis ribosome generated. It's resolved down to 3.7 angstroms. And so we're really excited to continue generating structures for French cell toleransis ribosomes, hopefully with these different homologs incorporated, translation initiation complexes, and be able to see at the molecular level what's happening when these ribosomes have different BS21 content. So at this point, I'm just going to briefly kind of summarize what I've told you so far, remind you our hypothesis is that incorporation of these distinct BS21 homologs may be producing specialized ribosomes, and this may lead to regulation of gene expression at the level of translation. And so what we've found is that we do in fact have heterogeneous ribosomes in French cell toleransis. And not only that, but these heterogeneous ribosomes appear to lead to alterations in phenotypes. Specifically, we see differences in gene expression, we see differences in virulence, and we see differences in susceptibility to antibiotics, which suggest to us changes in local structure or conformation. So there are many, many questions left to be addressed in this project. And so this is where, this is the early stage investigator aspect of this grant, right? Hopefully someday I'll be talking about the answers to these questions in another talk. But one of the questions we have is how the abundance of BS21 is impacting all of these phenotypes. For example, BS21-2 is the most abundant homolog in the cell. When we delete it, are the phenotypes we're seeing due to simply loss of BS21 or specifically loss of BS21-2? Right, so we are working on creating strains where we have a single homolog under control of an inducible promoter so that we can carefully control the amount of BS21 and match them among strains in order to specifically assess this question. How is this working at the molecular level, right? And so again, we are hypothesizing that BS21 is impacting translation initiation through interactions with the 5' UTR. And so we have some preliminary data suggesting that in fact there are specific sequences in the 5' UTR that may confer regulation by BS21, but that's still kind of work that's ongoing. And of course, so far we've really only looked at BS21-2 in Francisella tularensis, right? So what are the other BS21 homologs doing? Which genes are they controlling? How do they impact gene regulation? How do they all impact the structure of the ribosome? And I think one, kind of this is more of a future work, but something that we're really interested in following up on. This suggests to us, this raises the possibility that not only could we maybe design antibiotics that would target ribosomes for specific organisms, but we could also potentially create antibiotics that could target specific ribosome populations, right? So if BS21-2 really turns a ribosome into a virulence ribosome, can we inhibit specifically ribosomes containing BS21-2? And I would argue that our preliminary data suggests that that might be possible. So that's a really exciting future direction for us. So with that, I just want to thank the people involved. I managed to lack pictures, my apologies, but a huge thank you goes to my graduate student, Hannah Trotman. She generated essentially all of the data I showed you here today. So she's been a superstar in the lab. Tala, John, and Ollie are undergraduates who really pushed forward the antibiotic resistance project. Dr. Stephen Gregory serves as my mentor for this grant, and we have joint lab meetings every week with him in his lab, which are fruitful and enjoyable. Both of my departments have a joint appointment in the CMB and BPS department. My collaborators at Duke University, Dr. Marie Schumacher and her talented graduate student, Brady Travis, the proteomics resource that did the whole cell proteomics, as well as my funding from the Rhode Island Foundation and INBRE. So I'd be happy to take any questions.
Catherine, there's an awesome talk. We do have time for some questions, and there's two questions in the chat box. And if I have time, I'll ask one too.
Great. Yeah, so one of the questions is if other intracellular pathogens have multiple BS21 genes, that it might have different types of functions. So unfortunately, so I think that this observation about ribosome heterogeneity may not be applicable to every organism in the context of BS21, right? I think BS21, there's this kind of clear and obvious pathway forward because it's involved in translation initiation, but not lots of organisms have multiple BS21 homologs. Burkholderia pseudomallei is actually one of the ones that does, so that's particularly interesting to me. But I do think that the heterogeneity that comes from these multiple ribosomal protein paralogs is a rich source of potential regulation that has yet to be mined or really looked at very carefully. And so I'm really interested in seeing more about this. And so it's entirely possible that other intracellular pathogens have a similar, not exactly the same strategy, but maybe a related strategy. Hopefully that answered your question, Brett. Sujin asks if I expect kasugamycin will affect the BS21s differently. And I think our data suggest that it will. But unfortunately, remember I mentioned that abundance versus identity issue that we're having? Even in those strains with the isogenic strains where we have a single BS21, we thought we were doing our best to try to create cells that were very comparable, but unfortunately, they actually do incorporate different amounts of BS21 into the ribosome. And so I can't definitively answer that question yet, which is why I think it's so important to create these cells where we can titrate the amount of BS21 so we can answer that so we can interrogate that question really carefully. But I think that it might, right? We'll see.
We can
Okay.
we can leave my question for later. Maybe afterwards. I had a couple of questions for you.
Yeah. Well, thank you so much. Great. Stop sharing.
Our last speaker of the session is Brian LeBlanc from Brown University. And Brian, you can go ahead.
Okay, can you see my screen fully? Yeah.
Yes.
Okay. Okay, great, thank you. So, so I'm looking at the way, well, first of all, I'm part of the Center for Antimicrobial Resistance and Therapeutic Discovery, COBRE, at Rhode Island Hospital, and Project 2, which is looking at basically how we can modify our immune system to fight off fungal infections, specifically drug-resistant fungal infections. Next. Okay. So systemic candidiasis is a significant medical problem among critical ill patients. High morbidity, high mortality. It's prevalent among people with compromised immune systems. And antifungal resistance among Candida is becoming an increasing problem, whereas about 7% of Candida that is isolated from blood of infected individuals are resistant in some way to antifungal drug treatment. So our body's natural defense against candidiasis is the white blood cell neutrophil. It's the most abundant white blood cell in the body, and it's basically the Marines of your immune system. It's the first one in to tackle these infections. So here we have a nice little video of neutrophils attacking fungal hyphae. Now, the Candida uses, it's a transition to hyphae as a way to prevent itself from being cleared from the body. The neutrophil is much smaller than the hyphae. And what happens is it cannot phagocytose a hyphae, it's too large. So when it recognizes the hyphae, it sort of goes over and spreads out and does its best to kill the hyphae, but it's far too large to clear. So it relies on the release of antimicrobial molecules in order to defend against Candida. And upon recognition of Candida, that's how it becomes activated to release those molecules. So just looking at the cell wall of Candida itself, it is a complex cell wall containing a bunch of different components where you have an outer wall of mannan and proteins, and underneath that you have your glucans and chitin. And neutrophils have molecules on its surface that can recognize these fungal molecules, specifically an integrin called CR3, or complement receptor 3, is it's a unique receptor in that it can actually bind to two specific ligands. It has an I domain that can bind to actually a lot of different things. It's very promiscuous, but in terms of this talk, its binding to extracellular matrices of importance, specifically of fibrinogen. And then it has a lectin-like domain that can bind to the glucans. So it can actually recognize the glucans on the surface of the fungus of Candida and recognize that it's foreign and start its defense. One thing of interest, let me go back, is this outer wall of mannan. And one of the ways that Candida can defend itself against the immune system is to hide itself from the glucans. So the glucans and chitins are really important to be seen. So the overarching arching hypothesis of my lab, or at least my field of study, is looking at neutrophil activity. This enhancing neutrophil activity is important in order to mount an efficient antifungal response. However, the neutrophil will not recognize, or will not be in a position to recognize, Candida without being also in the presence of extracellular matrix proteins. So this, it's important for the neutrophil to see fibronectin and Candida at the same time. And what I forgot to say is this molecule CR3 can bind things together in order to show an enhanced response. So a neutrophil, when it responds to a fungal infection, releases all these intracellular granules and releases antimicrobial molecules. Specifically, what I'm looking at presently is this cute little project that I'm working on, is the chitinase chitotriosidase. And this is an enzyme that breaks down chitin. The mammalian system does not express chitin, so the only way it will recognize chitin is through a foreign pathogen. So it's interesting to see what this role is in clearing of Candida. And it seems to be important because chitotriosidase is present in all three granules of the neutrophil. And there seems to be a link between chitin and virulence. So if you basically modify chitin out of Candida, you attenuate its virulence. So there seems to be a link there. And when you look at the chitin content of different Candida subspecies, you see that some of the more, infective or Candida have higher chitin content. So the first thing my lab did was check to see if the neutrophil will release chitotriosidase upon recognition of Candida hyphae. So we put Candida hyphae in a well and we added neutrophils. And we have a molecule that is inherently fluorescent if chitotriosidase enzyme breaks down. So it's a sort of a fluorescent molecule that is attached to chitin that when it's cleaved, it becomes fluorescent. So as you can see, we see an increase in fluorescence with hyphae plus neutrophils. And if we use a chitotriosidase inhibitor called bisdionin C, we can block the formation of this fluorescence. So it's blocking the enzyme function. So there seems to be chitotriosidase released from the neutrophil. So we were lucky enough to collaborate with the lab at Brown that has a chitotriosidase knockout mice. And so we were able to take these mice and infect them with Candida albicans systemically, and then see what happens over time. And what was surprising to me in the beginning was over time, you have a normal response to the fungal infection is to lose weight as the fungal infection takes hold. And this is the weight loss of the wild type animal over the 5 days of the experiment. But our chitotriosidase knockout mice seem to retain body weight. Some actually had body weight above what they started with. So that was very concerning to me because I really thought that losing chitotriosidase would have made the infection greater since it's losing a defense mechanism. So after 5 days, I harvested the organs and I looked at the amount of fungal burden in the kidney, and the wild type has about a little less than 10 to the 7th CFUs of Candida, and we had a significant decrease in the fungal burden of mice that are deficient in chitotriosidase. And actually, we had 2 mice that pretty much cleared the infection. And so along with the decreased fungal burden, they also had a much decreased histology score. We looked at the kidney using H&E stain and scored it to inflammatory cells, inflammatory abscess, tissue injury, and hemorrhage. And in each case, there was a significant protection, it seems. There was less inflammation in these kidneys compared to the wild type. So it seems that the loss of this enzyme is beneficial to the host. So that was very surprising. How is it that you lose an enzyme that breaks down chitin, which is a structural component of the cell wall of Candida? How is it that losing that enzyme you do better? And that was a very hard question to answer. So I thought about it for a while, and just looking at the way the enzyme works. So here's the chitin molecule. It's a polymer of N-acetyl-D-glucosamine. And when it binds, it cleaves chitin and releases a dimer of NADG. And it just keeps repeating. And so over time, you start to get an increased concentration of what's called chitobiose, which is that dimer. And this is one of those things that reminds you that it's really important when you get your PhD to do all the reading that was required of you, because my graduate work was on CR3. And one of the things I remembered was NADG seems to also bind to that lectin site of CR3 in some papers from the late 80s and early 90s. So potentially, this chitobiose would have a way of interfering with normal binding of beta-glucan on CR3, affecting the neutrophil's response to a fungal infection. So one of the assays we have in my laboratory is a neutrophil clustering. It's a migration assay. And in order to simplify things, we sort of make a very simplified hyphae, where we take beta-glucan and immobilize it to the bottom of a well, and we add fibronectin to give that dual ligation of CR3. And when we add neutrophils, they cluster and they form these aggregates, sort of like what we saw with the neutrophils going after the Candida hyphae from an earlier slide. However, when we add chitobiose, these clusters are much smaller. And actually, I just realized I'm sort of highlighting the wrong. Can you see my cursor now? Okay, I'm highlighting it on a different screen. I apologize. These clusters are much smaller compared to the clusters you see on the fibronectin beta-glucan, and quantitation shows that it's a significant decrease. So it looks like this chitobiose actually does interfere with our CR3 response of the neutrophil. So that was against a simplified hyphae. So then we looked at what happens when you add true hyphae. And so hyphae and neutrophils form these clusters. When you add chitobiose, you lose the amount of neutrophils that are responding to the hyphae. The clusters aren't as big. And when we block chitotriosidase activity using bisdionin C, you see these superclusters. So there's a much more exaggerated response, and here it is in graphic form. So how does that play to yeast viability? Well, when we look at yeast viability using the reduction of MTT, we see that when you add chitobiose, you actually decrease the neutrophils killing of yeast. And when we add the bisdionin C to inhibit chitotriosidase, we actually increase the effectiveness of the neutrophil to kill the Candida. So there seems to be something about this chitobiose that is affecting neutrophils' function. So how do I go about determining if this affects the lectin-like site of CR3, which is a very difficult thing to do? So what I used was an antibody that binds to the lectin site of CR3. And as a control, I used an antibody that binds to the I domain. And when I added chitobios, I wanted to see if it would interfere with the binding of that antibody. So here we have the binding of antibody alone. And when I add beta glucan, since it binds to the lectin site, it decreases the binding of the antibody to the lectin site. So that was our positive control. But when I added the chitobiosin antibody, it blocked it as much as the beta glucan. So it seems that there seems to be an interference of the lectin site by chitobios. And when I used the positive control, which is on the I domain, there was no change in either glucan or chitobios. So that was my way of sort of linking the CR3 lectin site as a method or a pathway of interference. I have ongoing experiments now using genetic modified cells, neutrophil cell lines that are deficient in the ectin site. And we're working on that now. I don't have any data to share, unfortunately. And to see if it's specific for CR3 and not other glucan receptors, I used a blocking antibody against dectin, which is also a glucan binding receptor on neutrophils. And there doesn't seem to be a change in the clustering if I block dectin. And if I add soluble beta glucan, if I outcompete the chitobios with extra beta glucan, I'm able to recover the neutrophil response. So that is my way of linking everything to that lectin site. So if chitobios is actually the reason that the neutrophil is not as efficient, then adding chitobios into the animal during a fungal infection would significantly decrease its effectiveness. And so that's what I did. So I treated the animal with either chitobios or lactose as my sugar control and injected Candida for a systemic infection. And as you can see, they lost weight just like the wild type. However, I had a drastic increase in fungal burden in the kidney, increased pathology across the board, to the point where here's our wild type fungal infection. You see the same abscesses that you saw earlier in the wild type, but the chitobios, the kidneys were just, they were destroyed, basically. It was a very hard infection for them. And you can see a lot of pathology. So that sort of linked everything together. So the next step would be to, instead of using the knockout animals, is to use a chitotriosidase inhibitor in vivo. Now, unfortunately, what I've been using before is bisdionin C. Unfortunately, that is it's only soluble in DMSO, and the amount of DMSO I would need in order to treat an animal was far beyond anything it could handle. So I needed to look at other chitotriosidase inhibitors, and this is very exciting to be after our last speaker. And as Dr. Ramsey has introduced, we have kasugamycin. So kasugamycin is well, it's an antibiotic, and it has some antifungal activities. However, it turns out it has a very strong chitotriosidase inhibition functions. And when we added kasugamycin to our fluorescent assay, it significantly decreased the activity of chitotriosidase, actually an order of magnitude more than our bisdionin C. So we're really excited to be able to use this in vivo. It's soluble in saline, and it was a very nice addition to our lab. And so adding it into our clustering assay, we found that sure enough, we got an increased clustering response using kasugamycin compared to saline alone. So that was very exciting for us. It seems to work the same way as our Bisdionin C did. The question that we had was, well, does it have antifungal effects against Candida? It is an antifungal, but looking at the papers from the 1960s, it had no activity against Candida. And sure enough, we reproduced that in the lab, and we showed that it does not have any antifungal activity against Candida, even at high concentrations. Here we have fluconazole is active at 0.25, and then using a drug-resistant strain of Candida, it doesn't affect the viability of Candida with kasugamycin. So we treated the animals with kasugamycin and then infected them with Candida, and it was great. It did a great job of reducing the fungal burden, and the results that I just got this weekend that I didn't include in the talk was we infected animals with this drug-resistant, this fluconazole-resistant strain of Candida, and we found that it actually had a stronger response against the drug-resistant strain than the wild-type strain. So we're really excited about using kasugamycin as a therapeutic, at least to help fight off drug-resistant Candida. So in summary, chitotriosidase deficiency, surprisingly, resulted in decreased fungal burden, decreased pathology scores, decreased inflammation overall, and the effect seems to be the creation of this chitobios that inhibits neutrophil activity, the chemotaxis, the fungal killing, and it seems to work through the lectin-like domain. However, I'm doing experiments now to address that. And then our friend kasugamycin also seems to work really well. It enhances clustering activity and increases candidal clearance. So obviously I didn't do these work myself. We have a really nice lab here at the hospital. This work in the beginning was done with my assistant, Nick, and is now currently being done with Amrita. I have a postdoc, Ritesh, who's really just done a great job of doing the in vivo work. I get a lot of support from the other members of my department in the Division of Surgical Research. My old mentor from PhD is Jonathan Reichner. He still helps me out. He's a great collaborator to talk to. Craig Lafort helps out Dr. Ayala. And also the members of the COBRE are fantastic to talk to, Dr. Now and Dr. Bobinchuk, and Dr. Liette Brown, who provided the chitinase knockout mice. So I'm happy to answer any questions.
So we are at the end of our session, and obviously, as we've kept saying, we'd love for everyone to stick around and ask our speakers questions because we tended to go over a little bit, but I just wanted to thank everyone for participating, and also to the audience. I think it's been a really interesting session where we found lots of common threads across all of these different talks. And it's been a pleasure to organize this with Sally, especially because I think a lot of us would not necessarily find ourselves at the same conferences. And so I hope this will lead to some interesting conversations. And again, thank you everyone. And please stick around if you're interested in chatting. I don't quite know how to organize this part, but probably we can just all talk to each other.
Yeah, that sounds good, and I'll start by thanking Sally and Sujin. This is great. Thanks for having us all here.
I have a question. It kind of is for Brian, but then I have a slightly altered one for Vicky as well. I just was thinking about, like, as I think about, like, in the news, reading about like these totally resistant fungal infections that, I don't know, it seems like they're more frequent, but also occasionally seeing things in the news about some of these parasitic infections, which are super scary. I just wondered if both of you could comment on, is there a change in the frequency of these infections? Maybe like for Brian, does, like, you know, is there something changing about our population that's making us more susceptible to these fungal infections? And then the other thing I was thinking about for Vicky too was, you know, is like things like climate change. Are those important, you know, for frequencies of infections? I don't know what your thoughts are.
Can I speak first? So in terms of frequency of infection, it really depends on you know, the target population that is susceptible for infections. Cancer therapeutics, the more people that are on chemotherapy, the higher the potential that there's going to be. Organ transplants, the better science is becoming, the more people are becoming immunocompromised. You know, we're getting really good at, you know, doing a lot of fun things with science. Now, specifically COVID, that is a major one that I'm concerned about because it is a respiratory infection, and there's a potential for there to be respiratory problems. And I'm looking at India, and I'm looking at the black mold that's going on there. That seems to be a major thing on the horizon that we have to address. But more importantly is the acquisition of these drug-resistant strains. Every year, we're getting more and more little places that are having these drug-resistant strains becoming a problem. So as time goes on, this is going to be a major problem. And this is actually why our COBRI was created, was to address that problem. And hopefully we can find novel ways of treating infections, treating fungal infections. Yeah, you are immunocompromised, but you're not deficient in all your neutrophils. Let's just make those neutrophils that you still have more effective. And that's what I'm hoping to do. And hopefully the treatments that I come up with in my lab are able to overcome and to give you that boost. You'll always have antibiotics. They do something. And hopefully together you're able to clear those things.
So I probably am not super qualified to comment on antimicrobial resistance in toxo, because simply we just don't see it in toxoplasma infection, and that's for a very obvious reason, that the cycle is not completed. There's no transmission from human to vector to human, or no direct transmission from human to human, so we don't get that selection that we see in something like malaria, where of course drug resistance is a huge ongoing problem and probably will always be a problem. We're just going to have to keep coming up with inhibitors for malaria. And I think it's, you know, I don't think it's going to be ever solved. We just always have to keep one step ahead of the parasite. And there are some really nice ways that you can look at fitness of the parasite after it develops drug resistance in Plasmodium, and, you know, come up with a way to kind of counteract that to make the parasite susceptible to the original treatment. And some of those approaches have been effective in the lab, but not so much in the clinic. So I don't know. As a toxoplasma person, I'm not super qualified to comment on malaria. I'm afraid to say anything that might be wrong. So, and how climate change would affect that, possibly, I think certainly climate change would affect prevalence of something like malaria for sure. As soon as those vectors, the mosquitoes, start moving out of their normal endemic regions, you're definitely going to get more transmission of malaria. Mm-hmm.
So that's interesting. So you don't see evolution of resistance in toxoplasma? Because
no, because
I guess
we're
not part of the natural cycle. Like, right?
So humans are dead end hosts. So the normal cycle for toxo would be for a mouse to be eaten by a cat, and then the cat becomes infected and poops out more oocysts, which the mouse eats, becomes infected, and then the cat eats the mouse. And in general, people are not being eaten by cats too often, so we're not. It's possible, of course, but I could certainly see drug resistance maybe evolving if we start treating cats with the same drugs that we treat humans with. I guess that's possible. It's just not prevalent enough, I guess, to really start becoming a problem.
No, they're not in farms, for instance, where they use.
Yeah. So in farms, they don't treat for toxoplasma. They do treat for other infections that are related to toxo, but there's no treatment for toxoplasma in livestock. They don't worry too. It's a problem for in sheep herds. It causes spontaneous abortion in sheep herds, so that's an issue. But for infection in the meat, they're not too concerned about it, because if you cook the meat sufficiently, you kill those parasites. So there's no transmission to humans as long as you're not eating the raw meat. So they don't really treat herds to get rid of the infection.
But Vicky, isn't the same concern about immunocompromised status for fungal infections similar to toxo infections? I mean, it was a major infection during the HIV, the initial HIV epidemic.
Yeah, definitely. It's definitely one of the top secondary opportunistic infections in AIDS patients, for sure. I think with the development of PrEP and antiretrovirals, it's not so much of an issue anymore. But there are preventative drugs that people take that are vulnerable to activation of that acute stage of infection. And actually, you may remember a couple of years ago, Martin Shkreli was that hedge fund trader who bought the company who had the patent for the toxoplasma drug. It was actually Daraprim that people were taking to prevent activation of toxoplasma. In the news, they were calling it an AIDS drug, but of course, patients would take it to prevent toxoplasma.
I had a question about the BS21 and the ribosome structure. Can you actually see the binding site or where BS21 is in that ribosome structure?
So BS21 is pretty loosely associated. So it's not actually found even on all E. coli structures that we have. So there's a question actually about whether or not it interacts with the 5' UTR. And I haven't found a structure with a resolved long enough 5' UTR in the current databases to be able to answer that question, but there is biochemical evidence and genetic evidence that it does, but structural evidence is lacking. And in the ribosome structure that we have right now, we do not see resolution for it. And we think it might be because we need to form a translation initiation complex. Okay. So we're working on that.
Yeah. And then Daniel, I want to talk to you about rework at Dartmouth. So I'll grab you some other point. So the other question I had about that was, do you think that it's actually affecting mRNA stability? Because you can imagine where if you don't bind a ribosome that you are eventually just gotten rid of.
Yeah, that's one of the, it's kind of a key control. We're hoping to do some ribosome profiling, but alongside ribosome profiling, you need to know what RNAs are there. So you need to do RNA-seq, and we need to test when we see some of these changes. We need to potentially even perform net-seq or elongate, to identify which transcripts are actually being translated at any given time, because translation stabilizes transcripts, right? So this interplay of transcript abundance and translation, it's very intimately linked in bacteria. So yeah, it's a consideration for sure.
Okay, and then my other question was the ribosome profiling, because I was curious as to.
We're working on it. We've got, we're getting our polysome profiles. So we've got some beautiful polysome profiles, and we've identified BS21 in active fractions. And so it's on its way. I just need to recruit some more hands to get it done.
I had a similar questions. So do people know what's the difference between those three versions of BS21, or
No,
is
no
the
one studied these.
domain that binds it? Because I thought it was interesting that it both up and down regulates translation, right?
Well, you can imagine that it could promote
it's not
translation of
that much of a
certain
difference.
mRNAs, and then in the absence of it, other mRNAs might be preferentially translated, right? So it may not promote, it may not repress translation, but it may permit translation of specific mRNAs and, you know, be a little less conducive to translation of other mRNAs, right? But we don't know, and that's one of the exciting things to find out, right? Because no one has really looked at this very carefully. All of those other phenotypes I mentioned, you know, no one's looked at the molecular mechanism that underlies those phenotypes.
Because it's not that large, the effect, right? In either up or down.
It's subtle. It's more like, you know, like a small RNA effect, right? This isn't transcription where you're completely on or completely off. It's regulation, right?
Right.
So, yeah. Yes. Thanks for those questions. I'm excited about it. Can I ask a question about your antibiotic, the resistance mechanism that turns on? I was thinking about this, where you have the larger well and you have the antibiotic resistance genes turned on and persisting. And what's the relationship between the persistence and the slow growth? Do you think the persistence is only because those bacteria are growing slowly, or is there something else?
Okay,
Would
so,
any bacteria,
yeah, so that's very interesting, and I would have liked to talk more about it, but we're not even that sure about the causality there, because if you overexpress this resistance protein, like this efflux pump, it deserves member potential, so it can also reduce the growth from that end. But also, like, if the cells are already growing slower, they can express that can cause the cell to overexpress resistance for more than one reason. One is that because of the cell is growing slower, so all of the processes are going more slowly, right? So effectively, like, you have more drug coming in because the cell is acting more slower, right? So then just because the cell is growing slower and it's not growing and diluting the intracellular components, you actually have the concentration of this efflux pump actually stabilizes at a higher level. And also because of rearrangement of the cell metabolism, there's also some other reasons of why the cell ends up expressing more of these proteins that are not part of, like, the core of the metabolism. So it gets pretty complicated, but then you do see this overexpression of resistance in this slower growing cells. But then as far as that colony goes, right, what happens is that when you arrest the cells at the top, right, there's more food that comes from the bottom, comes to the bottom, right? So then the cell starts growing slower now, right? And then they start to overexpress resistance, right? And then they grow, the cells that express resistance grow, then they occupy the top of the colony, right? And now the top of the colony has resistance, right? So it starts to grow faster and consume more resources. So then those cells that are at the bottom that transiently grew a little bit because of this a temporary availability of nutrients, now they go back to dormancy after having this period of slow growth where they overexpress resistance, right? So then now they're dormant again, and they had this period of slow growth to express resistance, right? So then now they're stuck there, and if they take the drug away, they stay there because the boundary between the growing and non-growing cells has shifted back up. So
and they're still expressing the resistance gene. There's or no.
So they're not growing anymore, and they keep their levels of resistance that they that they manage to express during that transient growth. And actually, if we take this colony and we expose it to like cycles of drug, right? So you expose them to drug, take it away, expose to. Every time they go through one of these cycles, they build like more and more resistant at that layer there. It's pretty interesting.
So like in a nutshell, like the gradients are promoting maintenance of that population that's more resistant.
So this gradient, they create like this diversity, right? So it basically implements this mechanism of like, you know, each drug exposure primes the colony to resist more, like in a subsequent exposure, through making the cells in the back grow like only for a little bit and express resistance.
Daniel, you're doing those experiments with clonal like populations, right? Do you do competition experiments? Like if you have strains that have variation in resistance, and what happens to the structure of the populations within those devices?
So we do quite a bit of competition assays in the continuous culture devices, right, in our chemostats. So now we've been looking at the last part of the talk there on the Pseudomonas populations, because we want to see how those populations evolve in the lung. So we want to see like what kind of conditions favor this emergence of these mutants that we see in the lung all the time. So so we do those kinds of experiments where we have the wild type and the mutant, and then kind of try to play with the conditions to see, to see what favors the emergence of these mutants that we see all the time. And a lot of it has to do with like having like a dynamic environment where conditions change. Yeah.
Any lingering questions? No, we're all good.
So, Sally, I asked the question about I noticed there were a couple of helix turn helix DNA binding proteins that you had annotated in that prophage. That's kind of why I asked. I wasn't sure if they were legitimate transcription factors, but I was also thinking, you don't need much of a transcription factor to be functional, right? You don't need high expression, so any of them might be interesting.
No, and I haven't tested those genes yet, and I went after the one that was most interesting to us first. But I actually think there's another set of genes that has a couple of membrane proteins. It wasn't really on that right arm, but that was kind of inserted into close to where the structural genes are. And it has a couple of membrane proteins in it. And so I'm sort of interested in those as well as possibly altering the ability of the drugs to get inside the cell. So we're having trouble cloning those yet. So we haven't gotten after that one yet. But yeah, there's a couple of at least DNA binding proteins, and I have no idea what they do, and they're adjacent to other genes that I can't predict any function for them. So
cool.
So Gina, I also have a question. I know it's not the main part of your talk, but how does the motility mechanism of Dicty works?
It's just they have, pseudopods. It's amoeba, so they just move and engulf prey. And
so do they move over each other, or do they just
when they enter the social cycle,
the slug part.
Yeah, so they I think the best way to describe it is that they basically glom onto each other, secrete an extracellular matrix, and then as they move in the slug, they're swirling on the inside and like causing it to migrate.
But the individual cells do move in relation to each other. They don't move as a block, right?
They do move as a multicellular body, but yeah, it's caused by the individuals all responding to whatever signal is occurring to cause the mortality. yeah, they're fun.
Yeah.
I know I've asked you this before, but so I'll just say I'd be super interested to see, you know, what's the difference in terms of those symbionts that are able to establish themselves better in the macrophage. It would be super cool to see what kinds of, what kind of genetic differences there are that allow them to be more established in macrophage. But the other thing I was wondering, I mean, I was trying to follow it because you had like 3 different dicties, right? And the different symbionts with them. And there was that one that was able to establish, a larger load in the Dictyostelium. And I just sort of wondered, and that one, I wondered whether you thought that the difference in fitness between the dicties carrying the different symbionts, is that like, is the resistance to some of the symbionts like a cost in fitness, or is it causing a difference in fitness? Does that make sense?
I think so. So
I said it started to make less sense as it came
well,
out.
I think it'll make a lot more sense actually to everyone, including myself, once I get the native hosts in there, because I expect they must be different in some way, that they don't seem to suffer the consequences of having these infections as much as anybody, any dicty that we find without the infection. One of the things that I've been trying to figure out that's kind of related to what we've been talking about is whether there has to be some sort of difference amongst the symbiont strains in terms of like, if they're too virulent and they're killing all of their hosts, then you expect that you wouldn't see those strains as often compared to the ones that are less virulent and more easily transmissible. And so like, we probably actually at this point don't have enough wild isolates to be able to test these types of ideas about what kinds of strains of symbionts might be more successful when they're not. When it's hard to say, right? There's not necessarily a bacterium that's trying to get inside a host that they can live inside there, but some of the arguments for the commensal bacteria that we have is that like you're in this nice, safe environment where your food is being delivered to you, right? So like you don't have to worry about competition as much if you're going to be the one that's going to establish like yourself inside an intracellular niche. And so it's been kind of interesting to try to figure out like, are some of these patterns that we're seeing with like, I guess I was saying with like these social microbiomes and how that determines like who the hosts are interacting with, who the symbionts then come into contact with, like what that leads to in terms of the types of hosts that we find more often, the types of symbionts that we find more often, whether there are in fact any genetic differences, or is it really dependent on the environment that all of these guys in that makes it so that you have differential like fitness advantages depending on what type of environment you're in. So I think, it's kind of complicated, but I think it's exciting. And so, yeah, well, we'll see what happens. Like Catherine was saying, we're, early careers. So hopefully the next time you see me give a talk, we'll have more concrete things to say about what exactly are these genomic differences that are contributing to these variation.
And also, what's the scale of the copy number of these symbionts? I think I missed it.
Oh, the intracellular, like how many are in there?
Yeah.
Let's see, do I know this off the top of my head? I think it's like probably in the tens within each cell.
Okay, so they can like you can lose them in cell division then.
It must be.
It's not like so many of them that so there's a chance that in cell division you're gonna have a.
Yeah, so in some of the cases where when it's a newest infection that we establish in the lab, I think when the fruiting bodies become old, it seems like the Paraburkholderia inside have been proliferating so much that they actually burst the spores that the amoebas have turned into in the fruiting body. So I think there probably is cases where we're not readily seeing it as often as the cases where the Burk are more contained in post non-lethal levels, where we're just like not seeing those cases where they have been over proliferating and killing everybody. And I suspect that's one of the reasons why, when I was saying that when we repeat these cycles of multicellular cycles, the load tends to go down. It's probably because that is happening where they're just killing some of the hosts that they're getting into, depending on the, yeah. Well, I think we are probably well over. Oh, yeah.
I was thinking about, kind of like what fibro cholera and its phage, it actually, attacks the mammalian cells. So do you think it could be that these things are allowing the Dictyostelium that are infected to actually keep away other Dictyostelium that aren't infected, or sort of killing them off or something like that? Because I mean, you could, end up with a system like that where it's advantageous to you, to the bacteria that's in the host, to keep your host able to beat out hosts that aren't infected.
Yeah, so I definitely think that's possible. I've been trying to figure out a good way to test this. And so what I initially started out doing is, we know actually that Dicty without any symbionts also compete with each other, so that sometimes some of them are more successful in that social cycle. And so I was trying to combine the infection with this so that we can actually see if there was an effect of the infection itself. And it turns out that because when I'm doing the experiment, I'm using dilutions to estimate the concentrations of the hosts and the symbionts. It's really hard to know the initial ratios that I'm actually combining them at. So I try to get to 50-50 as close as possible, but it's noisy. And so at this point, I can't actually detect what that difference is, whether it's due to the infection itself that's killing these new hosts versus any errors that I'm introducing into the experiment. And so it'd be really cool to do some sort of live cell imaging where you have like a limited number of cells in these wells and see what actually happens to the different hosts when they're coming into contact with these guys, not necessarily in the social cycle, but yeah, I haven't figured out a way to do this at this point.
Maybe would using qPCR to try to assess the amounts of each of these more precisely, would that aid in trying to figure that out?
With the host,
Infection.
if you had different host genotypes, do you mean?
Well, yeah, I guess, yeah, I'm just trying to think of, you're saying that part of the issue was this kind of variability when you're trying to quantify the amounts. I'm just wondering if somehow quantification might become easier if you had qPCR to, you'd have to figure out which probe will go against which host, and yeah, it might be a bit headachy. Yeah.
Anyway, so thank you everyone so much for participating. We really do hope that we run into you in the future and in person. And good luck with all of your experiments and research. And yeah, I think we are happy to let you go. Sally, do you and I just want to stick around to debrief a little? And
yeah, that would be great. And I just want to thank everyone for sticking around and having conversations and discussions. It's the piece that's always missing during these sessions, when we don't really get to ask all the questions we want to ask and get to give you more long-winded answers. So thanks to everybody for coming and speaking today, and thank you to everybody for asking such great questions and being willing to stick around and have longer conversations. It was really great.
Thank you.
Thanks so much.
Thank you.
Bye.
Cheers.